in the past slide. And unfortunately, many of these can also accumulate in edible plant tissues and negatively affect human health, which we're learning more and more about all the time. So in my lab, we study soil microbial ecology as a way to try to overcome some of these challenges. If you're not familiar with soil microbial ecology, it's just the study of all the biota that live in soil and carry out a lot of important processes and interact very intimately with plants. And so in my lab, we're specifically interested in these beneficial plant-soil microbial relationships, especially those that occur in the rhizosphere. So we have a couple specific research thrusts in my lab. One of those is on biological control of pathogens, which we don't have time to talk about today, but I will be giving a seminar in the agronomy department in February, talking more about some of our work in this area if you're interested in. The other major thrust that we have is how to alleviate these stresses caused by soil contaminants on plants, and more specifically, how to keep heavy metals out of edible plants for human safety. So now I'll move into talking a little bit more specifically about heavy metals and metalloids. So heavy metals and metalloids, things like arsenic, are basically just broadly elements in the environment with high molecular weight. So some of these are needed by plants in very small compounds, things like copper, zinc, and iron. And others, like lead, cadmium, and arsenic, unfortunately, are not needed by plants for any known reason at this time, but they can get up into plants and accumulate. There are many sources that bring these heavy metals into our environment. So sources like this often get the most credit, like mines, and in urban environments, decades of using lead paint, lead gasoline, things like that can bring these into soils, but there are also places in the world where there's geogenic sources, just parent materials, or groundwater that can also be sources of heavy metal contaminants. And so the challenge with these heavy metals is that once they get into soil, basically they don't go away. They don't degrade over time like other organic contaminants. And so ideally, if you had a contaminated site, you could move somewhere else, but in many parts of the world, this is not an option. There are only small spaces to grow crops. And if we want to make cities more viable, again, for urban ag systems, we need to come up with other ways to deal with these. Okay, so reducing stress and preventing uptake, there's a number of ways to deal with that. So luckily, not all heavy metals that are in soil are actually bioavailable. So they're not equally toxic. They're not available in solution to be taken up by crop plants. And there's many factors that can influence this heavy metal bioavailability in soil. So a lot of the chemical properties have been much better studied in relation to this. So things like pH, the amount of organic matter, phosphates, things like that in the soil can influence heavy metal bioavailability. But we also know that soil and plant microbiomes can have a big influence in making metals more or less bioavailable. And so there's microbes at the root interface that can increase bioavailability of metals, which is great in a phytoremediation scheme if you're trying to use plants to take metals out of the environment, but there's also other microbes that can stabilize heavy metals in soil and keep them out of plants. And so we're more focused in that group. And so we have a number of projects underway, just really trying to understand what governs heavy metal bioavailability in soil, with the long-term goal of looking at how to reduce that. And so we're looking for just example studies, how contamination really alters soil microbiomes and their availability to influence bioavailability and toxicity. And also then how we're managing crops now, how does that alter soil microbiomes and their resilience against heavy metal contaminants as they come in. So again, many studies in this area, and one of those by Usama, a visiting scholar in Pakistan. We just got funded published or accepted in Frontiers in Microbiology, so you can look for that soon. We also have studies just looking at local soil amendments that we can use to alter soil properties and therefore reduce bioavailability and uptake in plants, like this study here that was done by a visiting student from Colombia. So the other major way we're looking at trying to keep heavy metals out of crops is through crop breeding, collaborating with plant breeders. So we know, just like many traits, there's a lot of variability within many crop lines, genotypes for different traits, including heavy metal uptake. And so one project that we've been working on now for about 12 years, this is a long-term effort in collaboration with Dr. Phil Simon, who's an ARS carrot breeder. We're looking at carrots that are better suited to local food systems, organic food systems. But in my lab, we've been very interested in many years in this idea of developing carrots that are better able at excluding heavy metals. And so this was a study Gabriella did a few years ago to identify some of these lines. Similarly, in our work in South America and Peru, we've done similar trials in quinoa, demonstrating that different quinoa genotypes and based on where they were developed vary in their heavy metal uptake. So the next step in terms of moving forward and breeding for things like that is to understand the mechanisms that are responsible for these differences in heavy metal uptake. So we know there's a lot, including interaction with root microbiomes that we're looking into. But the challenge is just retaining the nutritional quality of these crops. So one of my colleagues who's a spinach breeder says we need to make sure we don't turn spinach into lettuce by knocking out the uptake of metals, because again, some heavy metals like iron and zinc are actually really important for the nutritional profile of our plants. Then also, we still need to look at both factors. So while we know there's genetic variability in uptake, we still, when we go out, we find that soil is a much stronger predictor of what gets taken up into plants. So we need to be working with both approaches to solve the challenge. So other challenges, again, we need to learn more about the mechanisms so that we can really understand how to keep heavy metals out of plants. And another challenge is with just existing screening methods for metals. So there are a number of ways that now are available to quantify heavy metals in soil. You can use these simple XRF machines. You can get the surface soil like it's shown here. You can get soil samples deeper in the profile and pack them into little cups like this, which is really nice. For some metals, it's pretty highly accurate, like things like lead, but it is not, they don't work well for other contaminants like cadmium that are in very low concentrations. So you can also turn to digesting in acids using an ICP-MS approach, which can also help, especially with some of these contaminants that are present in very low concentrations. But with both of these techniques, what we generally see is total, or what we're quantifying is total heavy metals in soil and not necessarily the bioavailable fraction, what is actually bioavailable, which we're trying to stabilize. And so there are some less harsh extractants you can use with ICP-MS, but how really accurate they are is questionable. So in contrast, plants are a much better sentinel or indicator of what's actually bioavailable in the soil, because there's what's happening in soil, but then plants can also interact with their environment, for example, by releasing root exudates that could make heavy metals more bioavailable. So plants are a much better indicator, but they unfortunately often show few visible symptoms of actually plant stress. So it's not like a phenotype like nitrogen deficiency or phosphorus deficiency that you can regularly see. So instead, we have to turn to these wet chemical methods to quantify uptake and stress. And so while these are very highly accurate with ICP-MS, we can get really high accuracy down to parts per billion within a plant. These are very expensive. It's time consuming to do this, and it's destructive. So you only get it at like one time point. And then also you generate lots of toxic waste with this approach. So these are all these acidic digests that are hanging out in our basement lab until we can get rid of them soon. So we keep REM in business, I think, and pulling away all of our waste. So that brings me in then to this collaboration that we did, again, with the folks at the phenotyping facility to start to look at, can we do something new? Can we use hyperspectral imaging to predict or evaluate, detect heavy metal stress in our systems so that we can better advance our research? So if you're not familiar with hyperspectral imaging, this is something newer to me, but this is just the simultaneous detection of all of these continuous narrow wavelength bands in a broad spectral range. So we can only see the small visible light spectrum, but there's a lot of others like these near infrared and infrared regions that we can't see with just the human eye. And so by using hyperspectral imaging, you can quantify and take images at these different wavelengths. And it turns out these can be a very sensitive predictor of plant stress. So you can get, depending on the wavelengths, very minute changes in leaf pigmentation, mesophyll cell structure, as well as even water content for plants. So a lot of people are using these in drought. And so we know cadmium, well, we don't see with heavy metals really strong visible signatures. We know things like cadmium can negatively impact plant physiological processes like chlorophyll, or photosynthesis and respiration that we might be able to detect. So that leads us in then to this study, and this study was led by a former grad student in the lab, Maria Zaya, and she actually came here to Purdue because of another crop. It's not a crop that you see here in Indiana, but it's, in my opinion, one of the world's most important crops. So does anybody know what this is? Somebody, you're cacao, right? So this is a chocolate tree. So very important in Colombian economy. But unfortunately, there's a lot of cadmium contamination in soils in Colombia, and cadmium can get up into cacao, the seeds that make up chocolate. And so she got a fellowship through the Cacao Project, which one of their goals was to find ways to deal with cadmium contamination in their systems. And she came here to do a project. So we had a really good time going out and visiting a lot of cacao production systems in Colombia and learning more about the challenge. And we worked very hard to get cacao clones going here in the greenhouse and inoculate them with cadmium for her study. And finally, we got these images. These were the last images before the phenotyping facility shut down before the pandemic. So I'm very happy to Chris Hoagland for letting us get these in. He wasn't happy about it, but there they are, her pretty cacao plants subject to cadmium and not cadmium stress. But most of her work, because it took so much time to get going on cacao, we were instead focusing on leafy green crops. So leafy greens in general are just the crops that are the most vulnerable to heavy metal uptake. There's kind of a clear trend over time, always leafy greens take up the most. Then you have root crops, seeds like cacao, and then fruiting crops are generally the least susceptible. So Leafy greens can take up a lot of heavy metals at levels that are unsafe for human health, so it's a concern. And we know that different species vary in susceptibility as well as potential visible symptoms. And so for her experiment, we selected 2 very contrasting leafy green crops to see if we could use hyperspectral imaging again. So one of those we chose was basil. This is an herb that's very susceptible generally to heavy metal stress, and it fit within the confines of the AAPF at the time, as it was a taller plant. And then the other one we chose was kale. So kale is a brassica crop that tends to take up a lot more heavy metals than other leafy green crops. So we focused on cadmium as the heavy metal of choice in this study. And this is just because of all the heavy metals, really cadmium is the one that tends to be the most soluble and available in soil, so the most problematic to try to deal with. It's very toxic to plants, to microbes, to humans in very small concentrations. Again, you only need 1 parts per million sometimes to really have problems in uptake. And there's many potential sources for cadmium in our environment. So some places, again, geogenic sources. So for example, there are shale deposits in the Salinas Valley where there are high levels of cadmium. And so that's the folks we know out in California are working on that issue. They can also come from anthropogenic sources, things like batteries and mining, but in many places of the world, especially South America, contaminated phosphate fertilizers are one of the biggest sources. So again, soil amendments, we know that some soil amendments can reduce bioavailability of metals in soil. So we were interested in her study to add one of these in there to see if we could detect these subtle changes. And we decided to focus on biochar in this case, which is one that we've had success with, as well as many other people around the world in binding metals in soil. And so if you're not familiar with biochar, it's just a material that's produced by cooking organic residues in very high temperatures and low oxygen, and it creates this very stable organic matter. So while we know it works, though, really there's still a lot of variability in biochars based on what they're made of and their pyrolysis conditions and how effective they are in stabilizing heavy metals in soil. And then also, of course, soil type, there's an interaction there. So we still have lots of work to do in really optimizing these types of amendments to keep heavy metals out. So the objectives then of this study, first, we just wanted to see whether we can use hyperspectral imaging to detect heavy metal stress in these 2 very distinct leafy green crops. Next, we wanted to determine whether this locally sourced biochar could lower this stress and if we could actually detect that in our experiment using the hyperspectral imaging. And then lastly, we wanted to identify vegetative indices that we could use to detect changes. So when you do that hyperspectral imaging, you get massive amounts of data, all these different wavelengths. And so it's very difficult to sort through all of those. And so people often use these vegetative indices, which just use a couple of the spectra to detect changes in plants. So we conducted this experiment at Purdue's new Ag Alumni phenotyping facility over there in Lilly Hall, which is a great place to work. It's very highly controlled environment, so you can really dial down specifically to what you're interested in and know you're going to have really high control to be focusing on what you want. And so the plants were grown in an artificial soil. We would have loved to bring real soil into this facility, but there are some weight limitations and there can just be some challenges in bringing our mineral field soil into pots. And so we created this artificial soil using sand, soil, and a potting media. Next, we amended the pots with either 0, 5, 10, or 15 parts per million cadmium, and we allowed this to incubate for 30 days to facilitate adsorption onto the soil particles as much as we could to, again, mimic what might be happening in the field. Then we came in and amended half of the pots with this biochar, this local biochar at a rate of 3% volume by volume. And then we planted either basil or kale and allowed these plots to go for, I think, 62 days for basil, 84 for kale until they reached maturity. So plant measurements, we did a lot of ground truthing, which is really important in developing these technologies. So Maria came in, she used a spad meter to estimate leaf greenness or chlorophyll content, used a ruler to look at height, counted the number of leaves, things like that. And then we also imaged the plants bi-weekly using the hyperspectral camera. So both from the top view and the side view, we were curious which would be better at really detecting the stress. And we, at each one of those times and each camera collected 473 wave bands with a spectral resolution at 1.26 nanometers. And then we evaluated 14 different vegetative indices from all of that data that we collected. So some are more common, like the normalized difference vegetation index, which is often used as an indicator of chlorophyll content, as well as others like a heavy metal index that has been developed by some other folks working in rice. Next, we also wanted to see if we could look at the whole plant, changes within the plant. So not only are we getting all these images, but we're getting all these pixels all over the plant. And so we developed these color maps and reflectance graphs so that if we could see if we could find minute changes within the plant and how they were being stressed by the heavy metals. And so that up there is an example. We used NDVI in this case for that purpose. And then lastly, at harvest, we looked at elemental concentrations of cadmium, as well as total carbon and a range of micro and macronutrients using more traditional methods. So results. So in basil, as we expected, it was more susceptible to the cadmium. So we were able to visualize some slight changes or impacts of the heavy metals. We actually saw a slight increase at 5 milligrams per kilogram, which does not mean that the plants were actually benefiting from cadmium, but we think it was due to a phenomenon known as hormesis, where plants at low levels of stress actually try to outgrow that stress. We also saw some slight visible changes in basil based on the biochar amendment. Really, the biochar kind of slowed the basil development, a little bit later flowering and things like that. So the cadmium concentration increased in above ground biomass of both crops as expected. And so obviously the higher cadmium in soil, the higher cadmium in the plant. So this was great. This really meant that we had the control of what we wanted. More symptoms in basil, not so much in kale. And we saw this nice linear increase in stress. And so the biochar itself reduced cadmium uptake by a small amount. And actually the reduction, this is not shown here, but was stronger in kale than basil. And we think that's perhaps because kale just took up more overall. So in terms of the vegetative indices, we found a number of different VIs that were able to detect cadmium stress in basil, including many that were associated with this red edge, which is really important with hyperspectral imaging. It's a indicator, a very sensitive indicator of plant stress. And we also saw that though with basil, of course, we saw greater signatures at higher stress levels, but that it was only available, or we saw the most early within the growth of the plant. So the plants need to kind of overcome the stress. And the images taken from the top view were much stronger and greater resolution than the images from the side view. So cadmium symptoms and biochar amendments were much harder to detect in kale, but there was one that was valuable. This is the anthocyanin reflectance index that we could detect at the first time point and also then taken from the top view. And this was one that also worked in basil. And so we could conclude that really this anthocyanin reflectance index, this is the formula that you can use to detect that, was the most helpful in our study. And we think that could be for a number of different reasons. We know that heavy metals can influence the production of compounds like anthocyanins and carotenoids, which are very important in protecting plants from heavy metal stress. And they can also detect subtle changes in iron and zinc concentrations. So heavy metals often interfere with iron homeostasis, and we saw that, and then you often get an increase sometimes in zinc as the plant tries to protect itself. So the color maps generated using this normalized vegetation index looked a little bit like this. There's basil on the left and kale on the right at low and high and early and late in the season. And so overall, NDVI was actually not a very strong indicator of heavy metal stress in our systems. And we think maybe the plants weren't quite stressed enough. But when we really zoomed in and looked at all of these little pixels and things, we could start to pick up some very subtle changes, especially in kale, that we were able to detect, like around the leaf margins and things like that. And we think that might have been due to the fact that overall kale just had lower nitrogen relative to the basil. So in summary, we think that this was really powerful, that hyperspectral imaging can become a powerful tool to help researchers like our lab to develop new ways to deal with heavy metal contaminants. And we think with further optimization too, it could help us start to learn more about plant stress responses and these interactions with soil that are occurring. And that biochar has potential, but we certainly need more work to optimize that. And so if you want to learn more about this study, you can go to this paper that Maria published earlier this year in environmental pollution. Okay, so next, taking this a step farther, we wanted to see, can machine learning be used to classify plants according to safety thresholds? And so this work, was primarily led by Augusto Sousa, who was a former postdoc in the AAPF, who knows a whole lot more about machine learning than I do. I'm still learning about this, so I'll do my best to talk about it. But if you have tough questions, make sure you reach out to Augusto, not me. So machine learning, if you're not familiar with what it is, it's something that gives computers the ability to learn without being explicitly programmed. And we thought it would have an opportunity to go beyond just quantifying plant stress using hyperspectral imaging, but actually predicting the concentrations that we would see. And that also going a step further, applying more deep learning approaches could increase our accuracy of these models. And that by conducting our initial experiments under these very highly controlled systems, that we could increase the accuracy as well to develop better approaches. So we decided to, again, focus on safety thresholds for heavy metals and soil. So if you're not familiar with these are thresholds that are created by governments to regulate safety levels within heavy metals. And so the most commonly used worldwide are in the Codex Alimentarius, put out by the FAO and World Health Organization, which has set levels of heavy metals in our food. And these are generally set based on kind of what is reasonable within crops. Like they recently did a review of quinoa and set the level at 0.2 parts per million. So leafy vegetables, the standard through the World Health Organization right now is 0.2 parts per million. So that's what we decided to focus on in this study. If you didn't know, actually right now, the United States currently has no legal safety thresholds. So there aren't requirements. There are if you export your crops, but we know that's going to be changing. There's been a lot of things that are happening lately, especially around baby foods, where the FDA is currently reviewing all these standards and are expected to come out soon with much lower standards, especially for baby food at the even 15 to 30 parts per billion range, which is going to be very challenging to meet. So the objectives of this study: first, we just want to determine whether hyperspectral imaging can be used as a non-destructive method to classify above and below these safety thresholds. And then we wanted to compare different machine learning algorithms for their potential to optimize these models. And so we took all the data that we collected during Maria's experiment for this modeling approach. And we followed kind of a basic protocol like this, where we started first narrowing down wavelengths from 418.9 to 978.9 to reduce some of the wavelengths that had a lower signal to noise ratio. Next, we created 3 sets of spectral data for the model training process. And so one of those was just the original data with all wavelengths. The next was key wavelength speech features identified using the Relief F program. And then we combined both what we generated from the Relief F spectra with principal component analyses to reduce the dimensions and further identify specific wavelengths to work with. So next, training the classification models. We've evaluated 3 machine learning algorithms, specifically artificial neural networks, support vector machines, and ensemble learning. And for each one, we divided all the samples, 75% for training and then 25% for actually testing the models. Each model was generated and analyzed 10 times with different random data sets to validate it. And then with each trial and data type, we used 3 methods: the recall, precision, and F1 scores, to evaluate the performance of the cadmium classification models. And this is really important to make sure the models weren't overfitting the data. So results, we saw clear differences in the visible near infrared reflectance spectra, both above and below this 0.2 milligrams per kilogram critical threshold. We could see some changes in visible bands within 400 to 700 and the near infrared bands, so areas that were critical for chlorophyll, as well as changes in canopy structure. So we can conclude the cadmium did have, a much more focused level and impact on chlorophyll. And then using the Relief spectra, we were able to narrow down and find other specific wavelengths, including some that in that important red edge measurement that were also altered by the cadmium. And so again, point to changes in chlorophyll content. So the first 3 principal components were candidates for deeper evaluation. We found a number that were positively impacted by the cadmium and one that was negatively impacted. And then using that, we found that it was possible to differentiate between samples based on these PCA scores. However, there was nonlinearity in the data, so we moved into using these machine learning algorithms that could handle the nonlinearity and provide better clustering than you can see here in these principal component analyses. So the best model turned out, we decided that since focusing on false negatives, which is the chance that a plant will be considered healthy when it's actually contaminated, is more important than false positives. And so the recall metric was the most valuable then in evaluating our data sets. And the best overall model was the artificial neural networks, especially the one trained using principal component analysis. So in conclusion, we think that combining hyperspectral imaging and machine learning can be a very promising tool to help reduce food safety risks and help researchers design new ways to keep heavy metals out of crops. And that these technologies can also help us better understand mechanisms of how metals and other soil processes then are influencing plant physiological processes. And so this is a manuscript we've submitted. We just recently submitted a revised copy, so we're hoping this will be accepted for publication soon. So next step, where are we going? We want to keep using this and how we're going to keep moving forward in our research. So one, we want to keep moving along these lines, collaborating with our breeder colleagues to develop varieties that have lower heavy metal uptake. And so I have a wonderful new graduate student, Kathleen Sapp, who's carrying forward with some of our work associated with the Carrot Improvement for Organic Agriculture project. So a part of that, she's doing trials across the state on 10 urban farms. We're looking at these genotypes that we've evaluated that have high and low uptake, validating how well that can work in the field in soils that have very different soil conditions. And so we do see some really nice, somewhat this holds up in fields for some of our genotypes. So you can see the 3271, it has about half as much cadmium uptake as the 6220. But again, we see soil also plays a very important role. So moving forward with using the hyperspectral imaging technology, Kathleen's going to be looking at, can we actually detect differences among genotypes using this screening method so that we can start to evaluate and investigate mechanisms. So she did just a couple weeks ago a pilot experiment to learn how to grow carrots in the AAPF. And then just on Monday, she planted her first experiment with these genotypes at high and low cadmium concentrations. So she'll also be following up with some greenhouse studies to better tease out the mechanisms of soil genotype interactions using this benchtop unit that Young assures me is going to be here soon. Then we're also moving forward. We're continuing to collaborate with breeders. Phil is developing a mapping population with the high and low cadmium accumulating carrots that we want to see and evaluate using this imaging technology, because once you start getting into breeding populations, you get massive numbers of plants. So can this speed that process up? And then we're also working with some spinach breeders out at UC Davis with the hopes of getting some funding to start bringing their mapping populations here and evaluating them as well. So another important contaminant that we're interested in focusing on, so heavy metals aren't the only thing that happens in soil. Unfortunately, there are others, and one that is becoming more ubiquitous in the environment are these nano and microplastics. And so I'm very excited. Lele is a student at the University of Cattolica with Luigi Lucini, and they've been doing some work with microplastics. And she's here for 6 months doing an internship where we're going to try to dig a little deeper into what's happening with these plant soil microplastic relationships. So these are problematic. We know they can alter plant metabolism. So this is some of Lele's work where she's been looking at different types of microplastics and different rates and studying how they're altering the whole metabolome, in this case, lettuce. We also know that these microplastics and nanoplastics can be challenging because they can increase the movement of other contaminants in the soil, like heavy metals and organic compounds, as well as influence microbial processes. And that some, particularly the nanoplastics, have potential to get in plants. So the implications of that aren't totally clear, but we need to study and learn more. So she'll be initiating an experiment very soon in the AAPF. So next, contaminants of emergent concern. So this is, we have a student, Lily, who's working on this project where we've been working with 18 urban farms across the state to understand the soils in those systems and how their management practices are influencing their ability to deal with contaminants and protect their crops. And so we've conducted a range of soil physical, chemical, and biological tests on these soils, as well as plant and pathogen assays to understand relationships between contaminants and these other processes. And so luckily, in most cases, we see that farmers in the urban sites are reducing the total amounts, diluting in some ways these heavy metals, although there are still some outliers. But next, we want to say, well, what about some of these organic contaminants, things like PFAS? And so we're very excited to be collaborating with Dr. Linda Lee and Yoon Choi to quantify also a lot of these organic contaminants or contaminants of emerging concern in these systems. And so there's a need for more research. We don't have plans yet to use the AAPF facility, but working with Linda now to try to get some funding to move this forward. So lastly, moving to the field, it's, it's great. We can do all these experiments under very highly controlled conditions and work out these tools and things. We know that if we're really going to advance our efforts in breeding and understanding how amendments and things work with different soils, we need to get out to the field. And so, there's ways people are bringing hyperspectral imaging out to the field. It's using unmanned aerial vehicles to start to monitor biotic and abiotic stress. And this is something we've started to play with just a little bit. So this is an experiment more on our biocontrol aspect of our study, where we got the UAV from ICSC, Jason, that has both hyperspectral imaging and LiDAR capabilities. And so we're starting to try to bring that into our program, but still have a lot to learn. And we're also trying to get funding to then use this technology or see how we can perhaps use it in the field to detect heavy metal stress, or our capability to bind heavy metals using these tools. And so we think there's possibilities. There is a group recently that published a nice article using UAV technologies and this imaging, hyperspectral imaging, to actually map areas within brownfields using plants. And so in their study, interestingly, it was these pigment vegetative indices, just like we saw, that were most predictive. So lastly, I just want to acknowledge all of the people that have been a part of this research, helping to move it forward, especially Dr. Yang. I would encourage all of you to go out and meet with Yang if you're interested in learning how the AAPF can assist you in your research. I think, to me, it's been very valuable and a lot of opportunities to move us forward. I also want to acknowledge these folks, Kathy and David Brenner at the North Central Germplasm Repository in Ames, Iowa, that holds our nation's carrot and quinoa collections. I'm a member of that as part of our North Central Hatch project. And so they're a great resource to all of you as well, if you're interested in getting different plant material for your experiments. And of course, all the funding agencies, thanks to the College of Agriculture for supporting the AAPF, putting that in place so that people like myself could use that. All of the folks that have funded us, especially this group, the Healthy Babies Bright Futures Initiative. This was a great opportunity for me to work with industry. I haven't had a chance to work with industry a lot in my research. And so this is a collaboration between a lot of the baby food manufacturers in the United States who've been funding some of our work. And it was very good to talk to them and listen to them about their concerns. This is obviously a highly charged issue dealing with heavy metals. And so you have to be very careful about how you're doing it. And so I learned a lot from them. And so that's my last slide. And I'm happy to try to answer any questions if you have them.
A civil disobedience or unrest or whatever might happen in the room here. So she is ready for anything here. And so could someone check the queue, please, to see the chat and see and say, well, who has questions from the room here? Yes, please go ahead.
Yes. So you mentioned that you were able to better detect the cadmium uptake with the above plant images versus the side images. And that some of those changes were most apparent earlier. And so I was curious, like, are there certain like age leads or time points where it may be like most optimal for using this type of technology to pinpoint heavy metal contamination in plants?
And you
Yeah.
might briefly repeat the question.
Okay.
Yeah, you're better, I think, on this one than I am.
So the question was really around using like the top view versus the side view, and like when we were able to see plant stress, and you know, from early to late time point, and if we can use that somehow to kind of, I guess, further hone in our efforts in this area. And that's a great question. And that's really highly relevant too, because you know, there's a lot of leaf sensors too that are out there that maybe you don't need to take the whole plant image to really understand how to use this, especially when we go out into the field. And so I'd have to look. It was actually different between kale and basil. With the kale, I think it was the older leaves that where we saw this more than the younger leaves, and it was the opposite in basil. So I think we need to do more work to really answer that question. But yeah, it's a good one. Yeah.
So Laura, thanks for the wonderful presentation. So my question is related to agriculture and management in the local system. So you were talking about G by E, right? Genotypes by environment. I'm interested in the M, management. For example, what kind of management that can help, you know, farmers to alleviate or reduce the heavy metal stress? For example, in your biochar study, well, compost, other things can make a bigger difference?
Yeah, so the question is about urban agriculture in general, and not just G by E, but G by E by M, which of course, yes, is very important in urban ag systems. So, and I give a lot of talks to urban ag about how do you deal with this. And so there's many potential ways. You know, first of all, if your soils are highly contaminated, moving to raised bed systems is a lot better than trying to, probably more effective than actually altering the soil that you have. So you can put liners down. There are also people using just like barriers, like several feet barriers of wood chips, and then they bring in fill soil on top of that. Although I would caution that we have seen people bring in fill soil that's more contaminated than the soil present. So you have to make sure you test everything. Yes, there are management practices. We know both compost and biochar can both reduce the bioavailability, it dilutes things, you know, the plants too grow healthier, so it's diluted within their biomass as well. So within that all being said, we do have questions about how long that lasts. So for example, out in California in the Salinas Valley, they've been doing things just like raising pH, which I should have mentioned is another really important factor to reducing bioavailability. But now that's starting to break down and we don't know why. The pH is still there. I think, of course, it's all microbial communities that we need to study more, but yeah, those are some of the things. Yeah.
Very good questions. Any additional questions? Looks like, yep.
You mentioned using your machine learning process to
That was the one thing I
I know.
classify your hyperspectral images to say like high cadmium, low cadmium. Was that more of a you take all your data, you go back and process it later, or is that the kind of thing you could potentially run in real time while you're in the field?
Okay, so I think I can answer that question. Thank you. Okay, question was around machine learning. I was scared you were going to ask me about how it works, which I would not be able to do. But so the question was really how we did it and like really what the application was. So we were using it in our case to, see if we can go back and classify. So we took this huge data set and did these very, focused analyses to see, can we really separate it out? So now that we have that, we want to use that going forward. So we have now more specific wavelengths that we think are much more specific so that we hopefully can in the field actually use that to classify plants.
Okay.
Yeah, thanks.
There's a question online from Callis Simsic. What are your inputs for machine learning modeling? And your output and CD estimation?
Yeah, so yeah, the inputs, I'll try to answer another machine learning question about what are your inputs in the machine learning models, and yeah, the outputs. And so our inputs were really just all this massive amount of data that we collected. So all the wavelengths at every time point, the different pixels all over the plant. So that's what all went into it. And then, yeah, our output was trying to be that cadmium estimation.
So I'll ask, I guess this will be the final question. Are heavy metals a concern like in livestock grazing systems of like pastures and passing through the meat and milk and eggs and?
Yeah, so
then-
that's a great question.
Yeah.
Are heavy metals a problem in livestock systems? And the one thing that I have heard about and I'm aware of is for at some point, like in chicken production, they were using pesticides in somewhere. Maybe it wasn't a pesticide, it was something to help the chicken, but they had heavy metals in it, and that bioaccumulated in their manure, which was then being composted and brought to farms. So it was contaminated compost. So again, heavy metals never go away. They just kind of move through the food chain. So yeah, there's potential in livestock systems as well.
Yeah, and that was what I was concerned about, is sometimes they can intensify, in those types of systems. So
Yeah.
Let's give Dr. Hoagland another hand for a great job today. And you do a ton of work outside of this too, because I looked at your promotion document for your courtesy appointment, I must say. And I couldn't believe how much stuff that you were doing.
Yeah.
So you're very, very vital member of our university and college here. So thanks again. I
Thank you
appreciate
very
it.
much.
So, and thanks to everyone for attending today. And just the two upcoming in a couple of weeks. We have two more. We don't have the slide up here, but we have two more this semester, and then we'll begin it all again next spring. We're already starting to plan for that. So, okay, thanks.